Okay, so just a couple announcements before we get started today. So I know you're probably wondering about your tests. I'm planning on getting them back to you on Wednesday. Actually, I have quite a lot graded. I think all that's left to grade are the essay questions. Planning on getting those back to you Wednesday. There is a makeup that I have to give, but I don't think I should wait to hand them back till this student is back in class, because it looks like she might have to quarantine due to COVID. So I think I'll just have to give a separate test to that one student and get yours back in a timely manner. So I'm planning on Wednesday. Let me know. I'll let you know what's going on. The reason I'm kind of busy lately is we had to move kind of on short notice. So Friday, there were movers scheduled to come to the house, and they came earlier than we thought. So that's why I couldn't be here on Friday. But after Tuesday, I think we should be pretty much settled in. And hopefully I'll have enough time Tuesday to get those graded for you. So that's about it. Let's get back to, well, we have a little bit of chapter 11 to finish up. All I wanted to do as far as chapter 11 goes is to show you this video. Let me just set up the share and I'll, all right, why can't I see that? Well, that's not going to work. Give me just a second to get this set up for you. Okay, there we go. So again, when we left off on Wednesday, we were just about finished with chapter 11. We were talking about lectins. Okay, lectins are proteins that bind carbohydrates, and they're very important in cell adhesion and cell-cell communication as well. The type of lectin that I was talking about at the very end of class on Wednesday was a type of C lectin. That's, you know, capital C. C lectins are calcium-dependent lectins, meaning they need calcium in order to bind carbohydrates. Okay, specifically, the type of C lectin I was talking about is a selectin. Okay, selectin as in select, S-E-L-E-C-T-I-N. And we're just running out of time on Wednesday. I was explaining to you how selectins recruit leukocytes to the site of an infection. So I'm not going to go through that all again. I think this short video explains it very well. So let's have a look at this. Okay, I don't know if you're getting audio. I don't know if you're getting audio via Zoom, but we're not getting audio here. I think that's because I'm using the laptop audio. Audio from the computer. Hopefully the video doesn't echo like this. Okay, sorry about that. Oh, there we go. Okay, I'm going to try this one more time. Sorry about this.
Leukocytes are white blood cells that help fight infection.
Dr. Neri, we cannot hear you.
Okay, there we go. That's not better, is it? Okay, let me turn down the volume. Okay, how's it now? Good? Okay. All right, and like I was saying, so we have specific proteins that allow things through the plasma membrane. We have channels that generally allow ions through. We have carrier proteins that can transport something down their concentration gradient. And we have pumps that transport things against their concentration gradient. Membranes are sheet-like structures, only 2 molecules thick. In nanometers, that's between 6 and 10 nanometers, that form closed boundaries between different compartments. So that's actually pretty amazing. It's so we all know that membranes, well, we should know from general biology that membranes are phospholipid bilayers. Okay, bilayer meaning just 2 layers of phospholipids. So they're only 2 molecules thick. They form closed boundaries between different compartments. So, of course, there's the plasma membrane that separates the extracellular environment from the cytoplasm. And then in eukaryotes, we have organelles as well. So organelles create favorable microenvironments for different cell processes. So, for example, if we look at a mitochondrion, that's an organelle that's specialized for aerobic respiration. If we look at the endoplasmic reticulum, that's an organelle that's specialized for making primary modifications to proteins, including forming disulfide bonds, so it has to be an oxidative environment. Lysosome, okay, the lysosome is specialized for digestion of proteins, fats. So again, we have different microenvironments specialized for different biological processes, and these are all enclosed in a phospholipid bilayer or a membrane. Membranes consist mainly of lipids and proteins. The ratio is going to differ. I think generally about 50% protein, 50% lipid is where most membranes are. But it's going to differ depending on tissue. It's going to differ depending on specific organelles. It's going to differ by species. Different bacterial species have different compositions of protein and lipid. One example, if you look at the Schwann cells that surround neurons, they have a very low protein content. Okay, they're mostly lipid. Of course, their job is to insulate the axon of the neuron, so they don't really need proteins. They need a lot of lipid to act as insulation. The bacterium I worked on in graduate school, Haemophilus influenza, about 50% of the outer membrane was protein, the other 50% was lipid. So again, it's going to depend on species, it's going to depend on tissue, it's going to depend on organelle, exactly the protein to lipid concentration, composition of a membrane. Membranes also contain carbohydrates linked to lipids and proteins, glycoproteins and glycolipids. Okay, glycoproteins and glycolipids we find only on the outside of cell membranes. So that's what we're looking at here. If we look at this figure below, we can see the phospholipid bilayer of a cell membrane. So we have different types of proteins. We have channel proteins. Again, these channel proteins are for transporting ions across the phospholipid bilayer. Integral membrane proteins, we'll talk about these later. Integral membrane proteins span the entire bilayer. Glycoproteins are proteins that have lipids, they have carbohydrates attached to them. Glycolipids, here's a glycolipid here and here. Glycolipids are phospholipids that have carbohydrates attached. And again, in both the cases of glycoproteins, where the carbohydrates attached to protein, and glycolipids, we're only going to find the carbohydrate part on the outside of the cell. Again, one thing you might have heard me say again and again, especially in cell biology, is that cells are sugar coated. So the carbohydrates that are attached to the glycolipids and the glycoproteins are what make up what's called the sugar coat of a cell. Okay, now if we consider the phospholipid bilayer itself in the absence of any proteins, it is selectively permeable as well. Okay, and by selectively permeable, I mean that it will let some things pass through freely, but not others. Membranes have both hydrophilic and hydrophobic moieties, which makes them amphipathic. When we look at the structure of a phospholipid, we have a hydrophilic head, which are represented in this drawing by these round heads. And we have two hydrophobic hydrocarbon tails from two fatty acids. And they're in the center here, represented by these straight lines. So this makes a phospholipid amphipathic. Okay, amphipathic means having both hydrophobic and hydrophilic components. And the amphipathic nature of phospholipids is what allows them to spontaneously form these phospholipid bilayers that make up membranes. All right, so before I go on to the next slide, let's talk about the selective permeability of phospholipids. So molecules that are small, nonpolar, and uncharged are able to diffuse across phospholipid bilayers directly and easily. Okay, so some examples of molecules that are small, nonpolar, and uncharged. O2, molecular oxygen, CO2, carbon dioxide, also some molecules like N2, molecular nitrogen, H2, molecular hydrogen. Okay, what all of these have in common is that they're small, nonpolar, and uncharged. Being small, nonpolar, and uncharged means that they can diffuse across a phospholipid bilayer freely and directly. Benzene is a little bit bigger. However, it's also a very hydrophobic molecule. Only hydrophobic molecules can traverse this very hydrophobic environment made up by the hydrocarbon tails of the phospholipids. So benzene, although it's a little bit bigger, it's very hydrophobic, so it can diffuse directly across a phospholipid bilayer. Molecules like water and ethanol, these can cross a phospholipid bilayer, but not freely like these small, nonpolar, uncharged gases can. So water or ethanol, they're going to need some osmotic pressure in order to get across the bilayer. Okay, so these can cross, but not easily. We need some osmotic pressure to get these across. If we look at large polar molecules, okay, things like glucose, amino acids, nucleotides, these things cannot cross the phospholipid bilayer by simple diffusion. Okay, now, of course, we need to get glucose, amino acids, nucleotides. These are nutrients we need to get into the cell. However, they need to go through a specific transport protein in the cell membrane in order to cross the cell membrane. Okay, so again, amino acids, okay, of course, which are Zwitter ions at biological pH, cannot cross. Anything that carries a charge. Okay, now ions are very small. However, because they carry a charge, that makes them hydrophilic. And anything hydrophilic, again, we have a very hydrophobic environment that they can't traverse as as charged molecules. Pardon me, not charged molecules, as ions, because these ions are very hydrophilic and carry either a positive or a negative charge. Okay, so that's the selective permeability of phospholipid bilayers. Small, nonpolar, uncharged can cross freely. Larger hydrophobic molecules like benzene can also diffuse freely across the plasma membrane. Small polar can get across, but we need some osmotic pressure in order to get them across. Large polar like glucose, large charged like amino acids or nucleotides, ions, anything carrying a charge can't traverse this hydrophobic environment made up by the fatty acid tails, so they can't cross the phospholipid bilayer. Okay, so more common features of biological membranes. Specific proteins serve as pumps, channels, receptors, energy transducers, and enzymes. So again, depending on tissue, species, et cetera, about 50% of the membrane is going to be made up of proteins. These have special functions. One example here, so here's a sodium ion channel. So sodium ion channels are very specific just for sodium ions. So they'll only let sodium ions cross the phospholipid bilayer. Again, remember, a sodium ion can't diffuse by itself because it's an ion and it carries a charge. We have specific transporters like glucose. Again, remember, glucose can't cross the phospholipid bilayer on its own because it's large and polar. So we need a specific transporter in order to get that into the cell. Okay, oxygen, carbon dioxide, we don't need transporters because these can cross the phospholipid bilayer easily. Membranes are non-covalent assemblies held together by many non-covalent interactions. So in the center, in the so-called hydrophobic core, where all the fatty acid tails are, we have the so-called hydrophobic interactions and van der Waals interactions stabilizing that part. Okay, we have ionic interactions. We have hydrogen bonding between the hydrophilic heads and water molecules. Okay, and we have ionic interactions between the water itself and the charged heads, and between the proteins that make up the phospholipid bilayer as well. So we have many non-covalent interactions that help to stabilize phospholipid bilayers. Membranes are asymmetric. The inner leaflet differs in composition from the outer leaflet. So I mentioned before that all of the carbohydrates from glycolipids and glycoproteins are always going to be on the outside of the cell, okay, always. So this means that the phospholipid bilayer is asymmetric. All of the glycolipids are going to be in the outer leaflet, or the layer of phospholipids that faces the outside. Also, cell membranes are made up of many different types of phospholipids as well. Some of them are going to be mostly in the outer leaflet of the membrane. Some of them are going to be mostly on the inner leaflet. For example, phosphatidylcholine is a phospholipid that we find primarily in the outer leaflet of the plasma membrane. Okay, so by outer leaflet, I mean the one that faces the outside. So if we look at this plasma membrane, so all of the carbohydrates, let's just make this glucose transporter a glycoprotein and put some carbohydrates on it. Okay, so all of the glycolipids, all of the glycoproteins are going to be on the outside. So this would make this the outer leaflet of the plasma membrane, and that would make this the inner leaflet down here. So all glycolipids in the outer leaflet. Also, the outer leaflet is composed primarily of the phospholipid phosphatidylcholine. So we're going to find most phosphatidylcholines in the outer leaflet. Another type of phospholipid is phosphatidylserine. Okay, phosphatidylserine we find mostly in the inner leaflet of the plasma membrane. So again, the plasma membrane is asymmetrical. Okay, so meaning that the outer leaflet is going to differ from the inner leaflet. It's going to differ in phospholipid composition. It's also going to differ in glycolipid composition. Okay, all glycolipids in the outer leaflet, none in the inner leaflet. Okay, membranes are fluid structures. Lipids and proteins are free to diffuse laterally and rotate unless anchored. Rotation across the membrane flip-flop is rare. So we can see the different motions that phospholipids can undergo. So again, because these are non-covalent structures, they're held together by many non-covalent interactions, they're not static. They're not going to stay in place. They can diffuse laterally. So we can see, here's an example of lateral diffusion here. So they can kind of change places and move around laterally. They can rotate in place like this. And very rarely they can flip-flop. So in other words, one phospholipid can flip to the other leaflet. Now, this is a very rare motion for phospholipids. There's also flexion. So flexion, the phospholipid tails can kind of move a little bit like this as well and flex. The reason why flip-flop is such a rare occurrence is because, again, we have this very hydrophobic environment made up by the fatty acid tails here. So from the bottom of these polar heads to the bottom of these polar heads, we have this very hydrophobic environment. Now, the heads of the phospholipids, pardon me, are hydrophilic. Okay, they have a phosphate group, which is negatively charged. They also have some polar groups as well. So traversing, again, a hydrophobic moiety like the phospholipid head, it's going to be very thermodynamically unfavorable for it to cross this very hydrophobic environment made up by the fatty acid tails. So that's why this is such a rare occurrence. It's very thermodynamically unfavorable. Okay, continuing with common features of biological membranes, cell membranes are electrically polarized such that the inside is negative. This plays a role in transport, energy conversion, and excitability. So there's a very important membrane protein called the sodium-potassium pump that helps to set up this concentration gradient. Okay, with more sodium on the outside than the inside. Also not shown here, there's more potassium on the inside than the outside as well. So this sets up a concentration gradient. The concentration gradient is important because the energy stored in that concentration gradient can be used to transport things across the plasma membrane. And also this makes an electrochemical gradient as well. Because there's more sodium on the outside than the inside, this makes the inside of the cell negative with respect to the outside. This is especially important in neurons. So neurons are the cells of the nervous system that transmit electrical impulses to the brain and from the brain as well. These electrical impulses happen because of depolarization of the plasma membrane of neurons, which are the cells that carry electrical impulses. So this electrochemical and concentration gradient is extremely important in signal transmission in neurons, and it's important in other cells as well. It's important in, like I said, for transport of certain molecules into the cell. Glucose is one example. All right, so let's talk about fatty acids now. So fatty acids are key constituents of lipids. Fatty acids are long hydrocarbon chains of various lengths and degrees of unsaturation that terminate with carboxylic acid groups. Okay, so here are a couple examples. So again, here's the long hydrocarbon chain. So hopefully most of you are familiar with this type of notation from organic chemistry. So we don't always write out all of the carbons and hydrogens because that gets a little bit tedious. So anywhere there's a corner, it's assumed that there is a carbon there. Okay, and also we don't draw in the hydrogens, so it's also assumed at each of these corners that there's 2 hydrogens. All right, unless of course a double bond is drawn in. So here we have the hydrocarbon chain, and at one end we have a carboxyl functional group. Same thing down here. So we have a long hydrocarbon chain. In this case, we have 2 double bonds and then the carboxyl group here. Palmitic acid has 16 carbons and no double bonds. And the way that we denote that is 16:0. Okay, 16:0 means it's a 16 carbon fatty acid with no double bonds. The ionized form of palmitic acid is palmitate. So you guys should be kind of familiar with this notation before. So remember I told you that when we look at the amino acids, like aspartic acid, for example, when the carboxyl group of the side chain is protonated, we refer to it as aspartic acid. If that carboxyl functional group is deprotonated, then we refer to it as an aspartate. Okay, so the aspartate is the ion that's made when aspartic acid is deprotonated. We use the same sort of language when we talk about fatty acids as well. So here's palmitic acid would be the protonated form. When the carboxyl group loses its hydrogen and we get a negative charge, then it's palmitate rather than palmitic acid. All right. Okay, so let's try something here. So if palmitic acid has 16 carbons and no double bonds, and the notation for that is 16:0, how do we express oleic acid in this notation? So let's count up the carbons first. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. Okay, so oleic acid has 18 carbons and 1 double bond. So how would we express that using this notation here? Anybody? 18:1, exactly. All right. Now linoleic acid, I believe linoleic acid also has 18 carbons. So let's just count real quick. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, so 18 again. So obviously for linoleic acid, using that notation, we would describe linoleic acid 18:2 for linoleic acid. Good. Fatty acids vary in length, usually 14 to 24 carbons, with palmitic acid and stearic acid. Those are the 16 and 18 carbon fatty acids being the most common. Most biological systems contain even numbered chains. Fatty acids differ in degree of saturation. Double bonds are always in the cis configuration and separated by at least one methylene group. Okay, let's go to the, okay, let's not go to the board. Let me try to draw with the mouse. So if we look at the double bonds, so degree of saturation refers to how many double bonds a fatty acid has. Okay, one double bond in a fatty acid makes it an unsaturated fatty acid. If we have more than one double bond in a fatty acid chain, that makes it a polyunsaturated fatty acid. All of these double bonds have hydrogens in the cis configuration. Okay, what does that mean? Again, I'm going to try to draw with the mouse, which is hard sometimes, so forgive my artwork if I'm less than neat. So that means the hydrogens are here and here. Okay, this is the cis configuration. Okay, cis configuration means on the same side. Cis is on the same side. Always in biological systems, we're always going to have the double bonds in the cis configuration. The other way is called the trans configuration. So if across a double bond we have one hydrogen here and one hydrogen here, okay, that's what's called the trans configuration. This doesn't occur, the trans configuration doesn't occur in nature. And it's a bad thing. Years ago, you guys most of you might be too young to remember this. There was a lot of press, a lot of stuff in the news about trans fatty acids, okay, and how trans fatty acids are bad for you. So in order to make things like margarine, what they used to do years ago, now, of course, unsaturated fatty acids tend to be liquid at room temperature because of the double bonds, and saturated fatty acids tend to be solid at room temperature because of the lack of fatty acids. So what they would do to make margarine was basically to take liquid oil and to bubble hydrogen gas through it in order to saturate fatty acids and make them more solid. This would lead to formation of trans fatty acids. So we would get double bonds with the hydrogens in the trans configuration. Right? Now, these don't occur naturally in nature, yet we were eating them. And these have been linked to, back then, scientists at the time thought trans fatty acids, if incorporated into your cells, could distort the cell membranes. And also, I think they were implicated in cancers as well. Nowadays, the way that we make margarine and other butter substitutes, we do it in such a way that we don't get trans fatty acids. Again, I'm thinking back like maybe 15, 20 years ago, Oreos were one of the things that were notorious for having a lot of trans fats in them. Again, that's not a problem anymore. I don't think there's any foods out there that still have trans fatty acids. All right, but again, always on these double bonds, we're always going to find the hydrogens in the cis configuration. Okay, properties of fatty acids depend upon chain length. When chains are shorter and less saturated, then they're more liquid. If they're longer and more saturated, it makes them more solid. What do you think this says about the level of saturated fats in vegetable oil versus butter? Well, I kind of already answered that question on the last slide. So when fats are unsaturated, one thing that's not shown very well in this drawing is when we get a double bond in the hydrocarbon chain of a fatty acid, it puts a bend or a kink into that fatty acid chain. So these are still shown as straight chain, but actually these would be bent because of those double bonds. This causes steric interference when we try to pack these together tightly. On the other hand, unsaturated fatty acids are the types of fatty acids that we would find from animal sources. So in things like butter, lard, all of these would be straight chain fatty acids. Butter, lard, other sources of fat that come from animal sources tend to be solid at room temperature. Okay, this is because the straight chain fatty acids can pack together more efficiently without steric hindrance. This is going to make them solid at room temperature rather than liquid at room temperature. Okay, so shorter chain and less saturated means more liquid. If the chains are longer and more saturated, they're going to be more solid because they're going to pack together more efficiently. Okay, so getting back to plasma membranes, there are three major types of lipids in plasma membranes. We have phospholipids, glycolipids, and cholesterol. Okay, phospholipids are going to make up most of the lipids in cell membranes. So about 75% are going to be phospholipids, like this here. About 5% glycolipids. Again, glycolipids, notice they're only going to occur in the outer leaflet of the plasma membrane. And then we have cholesterol. Cholesterol is about 20%. Okay, cholesterol's, we actually see cholesterol inserted right into the phospholipid bilayer. Okay, so first, phospholipids. Phospholipids are abundant in all biological membranes. So again, about 75% of all of the lipids in a membrane are going to be phospholipids. Phospholipids have four components, one or more fatty acids. It would probably be more accurate to say one or two fatty acids because phospholipids are always going to have two fatty acids. Sphingolipids are always going to have one fatty acid. We also need a backbone to which the fatty acids are attached. This is going to be, in most cases, this is going to be glycerol. So it's going to be glycerol in phospholipids, sphingosine in glycolipids, also a phosphate group here in purple, and an alcohol attached to the phosphate. Typically, the alcohol is choline, serine, inositol, glycerol, or ethanol. I'm sorry, ethanolamine. Okay, so again, four components. Fatty acid. So we're going to have either one or two fatty acid tails. We're going to have a backbone made from either glycerol or sphingosine, a phosphate, and then we're going to have an alcohol group. In this case, the alcohol group is choline, making this a phosphatidylcholine, but this can also be serine, inositol, glycerol, or ethanolamine. Okay, in phosphoglycerides or glycerol containing phospholipids, fatty acids and phosphates are attached to the glycerol backbone through ester bonds. So this example that we were looking at on the last slide as well, this is a phosphoglyceride. So in a phosphoglyceride, the backbone is always going to be glycerol. And so glycerol molecule again, pardon my penmanship, but I'm going to try to draw with the mouse today. Okay, so this is basically what a glycerol molecule is. I don't want to draw in all the hydrogens, but a glycerol molecule has 3 hydroxyl groups, 1 on each carbon. And these hydroxyl groups on the glycerol can form ester bonds with the carboxyl groups on the fatty acids. So the carboxyl groups on the end, so again, if we look at a fatty acid, we have the hydrocarbon chain, and then on one end, we have a carboxyl group. Okay. So the carboxyl group of the fatty acids forms an ester bond with the hydroxyls on the glycerol backbone. Okay. So in a phosphoglyceride, we're always going to have, generally we're going to have 2 fatty acids linked to the glycerol backbone via ester bonds. And also generally, it's not shown in this particular figure, but generally in a phospholipid, you have 1 saturated fatty acid chain and 1 unsaturated fatty acid chain. In this one, they show 2 fatty acid chains, but generally there would be one of these 2 fatty acid chains would be unsaturated. Okay, and we're out of time, so I don't want to get into sphingolipids. We'll start sphingolipids on Wednesday. Okay, so just to recap, I'm planning on getting your tests back to you on Wednesday. We'll talk a little bit about them Wednesday morning. Other than that, I will see you guys later. And if there's no questions from Zoom, I'm going to stop the meeting here.